Okay, which are SRM basic concepts. Okay, like I said before, in this session, the goal is to give you all the elements and all the concepts to know what is SRM. So when something is not clear, you have to stop me, because in 1 hour you have to know everything. Espera, mantengo el d'això. Okay. Okay, so we keep calm, relaxed, and we go on. What we have seen until now, okay, in a very global way, is that we were going to use targeted proteomics when we have a hypothesis to test. In general, it's very well suited to measure few proteins in many samples, as opposed to in the shotgun approaches, and is considered a quantitation gold standard. So many times it's used to quantify proteins. Okay, so the workflow of this talk, first I'm going to tell you about the type of instrument that is necessary to do SRM. Then we're going to learn what is an SRM assay. We're going to see which peptides we want to use to monitor our proteins by SRM. And we're going to see about SRM verification, meaning, am I sure that I am measuring what I want to measure? And about SRM quantification. Am I quantifying correctly? Okay, so SRM is measured in triple quadruples type of instrument. This is one of the types of instruments that you saw the first day. This is like the skeleton inside one of these instruments. These are just pictures of triple quad type of instruments of different vendors. And this is how a triple quadruple mass spectrometer in this cartoon works. Okay, this is a quadruple, which is formed by these 4 rods, to positive to negative, in which we can apply a potential. And the goal of the quadruple is to filter by mass. Okay, this you already learned. Now we revise that what quadruple do is that they let go a specific mass. Through here, everything that is not set for the mass that we want to filter and reach the detector is going to collide into the rods. So, okay, so in quadruples, we filter by mass. When I say mass, it's mass to charge. Okay, so this is this real picture, and this is the cartoon, in which we have a first quadruple that we set just to pass the mass to charge that we want for a certain peptides. Then in the second quadruple that can be used as a collision cell, we break these peptides. And in the third quadruple, we're going to filter by the mass to charge of the fragments of the peptide. This is the basis of an SRM assay. Okay, we have a mix of peptides because we digested our sample. We set the first quadruple to filter by MS1. MS1, when I say MS1, is the mass of the precursor ion or the peptide. Okay, when I say precursor ion, I'm talking about the peptide ion, because it always has a charge. Then we break these peptides, and we tell the third quadruple to only let go the fragment that I want. So this pair of peptide and its fragment is what we call one transition. Okay, so we are measuring this transition, that is this fragment that comes from this peptide. That's this way of operate the instrument makes SRM high specific and highly sensitive. This, yes. Yes, we are going to see this. Okay? Okay, so.
Do I want to pass only, to filter and let pass only one peptide? Is also specific?
Yes.
Oh. I thought it was like a range.
Yeah. Depending on the complexity of your sample, more things are going to go in.
Okay.
So we are going to see that this happens. But the concept is that we set our quadruple for one mass to charge. Then we break, and then we set our third quadruple to another mass to charge. Okay, so over time, we see how these signals, this light pink would be this fragment. And then we set up this quadruple to filter another fragment without changing this quadruple. Okay, so over time, we see how each one of these lines represents one of these fragments that we let go, okay? And the important point of this slide is that we can see that each fragment is going to coelute at the same time because it is coming from the same peptide. In the chromatography, the peptides are passing by. So when we see this type of signal, that this would be intensity and this would be retention time, that is like this one inside the other, all eluting at the same time, most likely they are coming from the same peptide. So transitions from the same peptide coelute. And this is how an SRM signal looks like. Okay, so an SRM assay is the list of peptides and its transitions that we measure for a certain time and with the correct parameters in our instrument.
And you change the fragment by changing the potential of the...
Yes, but it's not like you do it, but you tell the instrument, I want to monitor. You just have to tell the instrument, apart from the parameters, the masses that you want in the Q1 and the mass to charge that you want in the Q1 and the mass to charge that you want in the Q3.
So the precursor mass is always the same, but the fragments differ. There are different fragments.
In this case, we first monitor this peptide with this fragment, same peptide with this other fragment. Okay. And then we go to this peptide.
From one protein?
Or another one.
Oh, okay.
Can be all from the same protein or not.
No, I mean the coeluting one. The one that coelute are always from one peptide.
Yes.
Okay.
Yes. This, this peptide will be in a different retention time than this one.
But the fragments are the same.
But all the fragments from one same peptide coelute.
In the same retention time?
Yes. This is the most important concept. Okay? So what you have seen until now, when we are talking about spectra or an MS2 spectra, is this. This is an MS2 spectra. You have Y ions, sometimes you also have B ions, whatever, in which you have intensity and mass to charge. And in this case, we have a Y series, the 11, 10, 7, and 6. The equivalent in SRM would be this. Okay? The intensity still corresponds to the intensity of an MS2 spectra, which would be fragmenting the peptide and looking at everything. Okay? So the intense ions are still the most intense ion. But here we have time now.
Because you have fixed your theta.
Yes, yes, because you want to see what is coeluting, because that means that this is coming from the same peptide. And the intensities correspond to the intensities that you have in an MS2 spectra. So
I don't understand why they are coeluting, because fragments could be different.
but they,
The same peptide.
yes, but they coelute in the chromatography.
Yeah, but independently of the fragment is bigger or shorter.
No, because what you are separating in your chromatography is the peptides. So the peptide goes inside the mass spectrometer at the same time. It doesn't matter, it is not fragmented yet. It goes inside the okay? And then you fragment and you detect it in that moment. It doesn't matter if the fragment is small or big or what. This is time. This is...
it's not the time that arrives to the detector, but the time that it's the dilution of the time to detector.
It's so fast. You know, now we're going to talk about, we're going to put this in time a little bit more clear. Okay, but this is important to distinguish, and always remember that this is time. Okay, well, here we had different places because there were different mass to charges. Okay, so this is, you know, how we acquire data in MS2. We're going to be revising this. This is our first quadruple in the filtering mode. We break this peptide, and now we filter for this specific fragment, and we get a signal that you think, okay, why do I get 2? Okay, well, sometimes happens that we get some interferences or whatever. The probability that 2 peptide fragments have the same mass and derive from the same isobaric peptide is quite low, but it exists. We have noise, we have, you know, other species, yeah. But we're gonna talk about this later. Okay, when we have this, means that each one of these signals belong to a different peptide, because if not, they would be coeluting.
The intensity reflects the abundance or the size of the fragments?
The abundance, that intensity, yeah. Okay, remember, this intensity here, the fact that this is more intense than this, is just because this fragment gives you a higher intensity.
Because it's more present.
Yeah, because when we fragment a peptide, the peptide has a certain abundance. The abundance is 1, but some ions are more intense than others, because this depends on the exact sequence. The same way that 2 peptides that come from one same protein, some have higher intensity than others, and they all come from the same protein and the same amount in our sample.
But the fact that they have lower intensity doesn't mean doesn't mean that there's less of them.
Well, there is less of these fragments. Some fragments we don't even see. We have 0, 8, 9 because we don't get that fragment, but that is not telling us the abundance of the peptide. But it's just the way that the peptide fragments. We never have an MS2 with all lines exactly the same. That doesn't happen.
There's one.
Yes.
It's not abundance also, it's just like that.
Yes.
It's not like the chromatography we saw the other day.
No.
Okay,
No, this has nothing to do. This is only reflecting the intensity of each one of these ions. And each peptide gives rise to different intensity ions.
But it's just because, depending on the sequence, these peptides have tendency to break in different ions, and these are the most probable ions or abundant ions.
Correct. Correct.
Then this intensity you will use for quantifying it.
This, yes.
So it's relative to abundance.
So?
It's relative to abundance. If you can, with this you can quantify.
Yes, for this exact peptide, yes. Yes, because each peptide behaves in a way. And we are going to talk about this also. Okay, so Sorry. Oops. Okay, so if now we would measure a second fragment of this same peptide, okay, we would get this signal that is co-eluting with this one in time. Therefore, we know that our peptide was not this one, but this one. Okay? Because when we measure this peptide and its fragment, so this transition, actually, instead of measuring like in a continuous way, we can measure in these points, we can like take measures where the points are. And then we have extra time. What do we do with this extra time? We can measure, for example, in this case, another fragment of the same peptide. We use this time that we have. Okay? And in this time, we would go, this is extremely fast and is impossible to put in an illustration. But we would go first to this point, then to this point, then to this point, then to this point. So we are measuring in time. But it's so fast that I cannot represent it in a slide. Okay? But the point here is that when we see the second fragment, now it's clear that this is our peptide.
Oh yeah, because they have the same rotation time.
Because they have the same retention time. And because I even, well, when I finish this, because maybe I answer you, when I finish, because we have this extra time, we can also use it to go to a second peptide and measure one fragment of this peptide and another fragment of this same peptide. Okay? Because this is a discontinuous sampling. So this is what we call the cycle time. Okay, I don't know if you have heard about cycle time. Okay, in SRM, the acquisition is sequential. Okay, and the cycle time is the time that we spend measuring everything we want to measure. And in this case, we can measure more than 4 transitions, but in this case, for the illustration, we are measuring 4 transitions. When we finish, we go back to measure the first one. So the time that we spend sampling all these different things is our cycle time.
So you have like 20 transitions or 50 or
yes.
Cycle times are very big.
Yes. So this second point here would come after measuring the rest of the things in our cycle time, and then we go back.
Yes, but you lose sensitivity depending on how many transitions you...
There is a number of transitions that you can measure. You cannot measure infinite transitions. And this all depends on your cycle time. And we are going to see this, because this is a very, very important point. Very important. So you need to understand the cycle time.
But it depends on the evolution time of the sample.
Mm-hmm. Okay, so any question on the cycle time?
Yeah, so if you have like 20 proteins to measure.
Yes.
Yeah? So it would be 20 measures?
Yes.
And then this would be a cycle, the
yes.
measurements.
Yes.
Of one peptide and one fragment of the peptide.
Usually, like we will see, you measure more than one peptide and more than one fragment, because if not, you have a lot of uncertainty.
What if you have just one unique peptide, and can you base this on just like if you have a very homologous protein and you just have one unique, and then you are lucky enough to see it, can you just base your ideas on
yes,
one?
You can do that. Okay, so up to here, you know, this is, if you understand up to here, you understand what is an SRM assay. But, I mean, we will be repeating the concepts. But if you have any question, you have to stop me. So then the second question is, okay, we saw examples like this one. They follow these proteins, etc., etc. They are interested in these interactors, but they are not measuring these proteins. They are measuring peptides. In SRM, we measure peptides. So the question that you first think, like you ask me, so which peptide do I measure now? Well, I don't care about the peptides. I care about my protein. The peptides is like the proxy for our protein abundance. Okay, so now we are going to talk about which peptides should I target. So the one thing, if you only remember one thing about which peptide you have to target, is that this peptide has to be unique to your protein. This is the fundamental. Okay, if you measure one peptide that belongs to two proteins, you don't know what the hell are you measuring. Okay?
If you don't have this unique peptide,
you cannot be sure that you are measuring your protein. You need to have one unique peptide. The peptides that you measure need to be unique. They can be unique for a protein group or for a specific isoform. Maybe you don't really care about the specific isoform, and knowing that is unique to that group is enough for you. But it has to represent your protein and only your protein. If not, what are you measuring?
Should this peptide precise? For example, if you find 10 amino acid sequences unique to this protein, should it read precisely? What happens if this peptide also under processing by other proteases or something? You know what I mean?
No.
So if one peptide, for example, 10 amino acids, unique to one protein, we found this peptide. But what happens if this peptide is also processed by an unknown protease, I mean, in natural mode? Then it can be only 6 peptides, 6 amino acids.
Yeah, but you are going to look for this best peptide to monitor, okay, in your mass spectrometry data. So you are going to see if when you process your sample, you find this peptide. If this peptide is cleaved, you are not going to find it.
Only from shotgun data you already have.
For example.
But the peptides that are unique normally they should be very long.
No, not not necessarily.
You can check, for example, different proteases and find, no, maybe if when you trypsin you don't find a unique peptide, you try with another one or no?
I mean, usually you find a unique peptide and not so difficult.
Yeah,
it is not so difficult. But you have to make sure. I mean, it can happen that your protein is super small. Okay, and there are always cases that are very difficult. But in general, you find your unique peptides for your protein and that's it. Don't worry too much. Okay, so there are a number of rules that are like general rules that have been described, you know, specifically when SRM started being used in the community, of which peptides are the best. These rules are pretty logical. Okay, you want one is that this peptide has good response in mass spectrometry. Can I see this peptide in mass spectrometry? Because sometimes the protein has, when we digest a protein, some peptides we see, some we don't. This is a clear, good ionization. The correct length also for the mass spectrometer. Tripeptides are always the peptides of choice. We want to avoid unpredicted modifications. For example, methionines and tryptophans tend to oxidize. So if a peptide oxidizes, we will have both forms if we only follow one or monitor one. Amino acids that have a tendency to get modified. If we have a serine that is getting phosphorylated, then we won't know what we are monitoring. Avoid sites where it's very likely that we have a miscleavage, or in general we don't want peptides with miscleavages because maybe some percentage of the peptide is with the miscleavage and some not. So these rules are, the logical rules that one would want for their ideal peptide. Sometimes this is not possible, because then you see what you have and you start ruling out all this and you don't have peptides. So what you need to keep in mind, at least make sure that it's unique. And then we want to make sure that what we monitor is what we want to monitor, so that this transition belongs to our peptide. So we need to ask our question, and this is a review was published in 2009 when people started using SRM, about how specific is my SRM assay. Like, be careful, because here we have 3 peptides. They are different sequences, and they give rise to a huge number of same fragments. They have the same mass. They will all pass through the 2 quadruples. So these are, and this is what gives rise to what we call interferences in SRM. So this is going to affect our identification. We may be thinking we are identifying our protein, and we are not identifying our protein. This is a real chromatogram from SRM here in our unit of a very complex sample, in which we monitor these masses in Q1 and in Q3, and, well, we get these signals. So sometimes things are not so easy. Sometimes, yeah, we see our signal and that's it. But this can happen. So we need to verify which one is our signal. So the most confident SRN signal validation is adding your heavy peptide inside the sample. Extremely important to keep in mind. Your same peptide with some heavy amino acids is going to behave just as your peptide in the chromatography and in the mass spectrometer. Okay, so it's going to coelute, have the same peak shape, have the same rank of transitions, or of those different intensities that we were seeing of the fragments, is going to behave just as your peptide. So it's your best standard, always. Okay, so we have these peptides. We are going to synthesize or order these peptides with a heavy amino acid. Okay, usually we order, we are working with tryptic peptides, so we order the arginine or lysine with heavy atoms, and this is going to be our best standard. So if we go back to this figure, and we were seeing here how the different fragments from the same peptide coelute, they will also coelute with our heavy standard. Okay, so the light and the heavy will be always coeluting. And this is our best validation. Then we are sure. So it's always, always recommended to put your heavy standard.
Just to, like I got it, but only to confirm, now if this were our spectrum view, then they would obviously be heavier on the mass charge versus time scale. But because we are talking in terms of retention time, they are exactly the same.
Exactly, because this is retention time. This is not mass. Because they don't have the same mass.
Exactly, yeah. But the kind of sense.
That's why we know that these signals come from the heavy and these ones come from the light. If not, we wouldn't know if we are measuring our standard or our endogenous. But because they have different mass, we can know which one is the heavy and which one is the light.
But you would also need to change these voltage settings on your quadruple.
Yes, but you actually don't go and change anything. We just add in your list now these other masses that are different. So your list of masses to monitor now is double.
When you use this, obviously when you have to synthesize the Harry standard, you need to make sure that this is the candidate peptide.
Yes.
Unique. How do you choose the unique? By silico? To looking at all the protein database to see that
yes.
only your
yes. Definitely. You have to make sure that your peptide is unique, and you do that in silico. You look in your database. If you're working with human, you can ask if is there any other human protein that has this little sequence.
And you need to make sure that it breaks down with that.
So if you don't have a heavy peptide, all you do is a relative quantification. With a heavy peptide, it's an absolute quantification.
That is also true. We will also explain that. Here is just the point that once you have your heavy peptide, you are sure that out of all those signals, which one is yours? Because what can happen also is that, imagine you don't have anything here and you only have this signal. Okay, one. You monitor your peptide and you get this. Say, great. Now you synthesize or you buy your heavy peptide and it happens to come out here. What is this telling you? That this was not your peptide, it was some interference and you were happily monitoring your peptide. Maybe it was I don't know, keratin, who knows. So the peptide, the heavy peptide gives you the security. Okay? So this is how the SRM would look. This is your endogenous or light peptide in the same retention time that your heavy reference. They coalute, like we have been saying. The order of the transitions, meaning the ranking of these fragments, is pretty similar, it's the same, okay? And once you have this, you have your heavy peptides, then you go ahead and make your SRM assay development.
Sorry, but the slide back. These chromatograms are going to be together, actually, not divided like this.
It depends. You can look at it in different ways.
They're not doing different injections. They're doing different.
No, but this is one same injection, but in the software in which you are visualizing this data, okay, you can look at it in different ways, and this is what you are going to do this afternoon. You're going to use this software. Ah, okay. Oh, maybe the slide is not perfect, but okay. So you can visualize it in different ways, and you will do that. Here, we ask the software, show me this peptide. And here, we ask the software, show me this peptide. Simply.
Just
that.
When you have some also a little interference in your heavy, you can also have that?
It's possible. This is a real, this is real data.
Okay.
This is real data, but the thing is that when you have your heavy peptide, you run it in different concentrations and you see how this signal gets more or less intense, while this one probably is going to remain the same. So with the heavy peptides, you can, you know, really be certain. Okay, so in the next session after the break, then we will go step by step how you would set up your method. Okay, here we are just setting these different concepts. You would have now your heavy peptides. You would go ahead and do your method in your instrument. Say that you already measured your samples, and now you look at your results. How do you verify that what you see is what you want to monitor? One parameter that you look at is the transition rank versus your MS2 library. The MS2 library is the group of MS2 spectra that you have from your peptides. So they know how they behave. They know these different relative intensities of their ions. and this has to match this relative intensity here. This is a measure, sometimes it's not exact, but it gives you a measure that this is correct.
But the peptide doesn't fractionate every time the same way.
No. Ah, what do you mean? Yes, it does fractionate every time the same one if you use the same instrument and the same settings. Okay?
The light, you also fractionate
The same. This is the nice thing. The heavy and the light will have the same pattern here and the same pattern here, because this has to correspond. It can vary if this has been acquired in one instrument and this is in another, which it happens very commonly, but still is more or less the same. It cannot, if here I see the Y6, 7, 10, and 11, and here I see the 8, the 9, and the 5, it's not the same peptide.
If you have a heavy peptide and the small fragments going to have a different mass as well, is that still okay or not?
I don't understand.
Like when you have a heavy peptide and cleave it, and the fragment that you are interested, the composition, is going to have a different mass.
Yes.
Can you still use it?
Yes, you always have different masses for the light than for the heavy in the fragment, but also in the peptide. Your Q1 is already different because the peptide is heavy. And if you don't have a different mass, you couldn't distinguish which signal comes from which.
I think the question is when you use the transition of the heavy fragment. From your heavy peptide, you have your arginine or lysine, which is the heavy one.
Yes.
But this one you don't include in your transition list.
They will be, because all the Y peptides will include that heavy lysine or heavy arginine.
You said, but you will select it then. Also.
Yes, of course, you have to. All the Ys from the light and from the heavy will have a different mass because they include that heavy amino acid. And we want them to have a different mass.
Okay, no, yes, you're right. No, I got it.
Okay?
Yes. But I don't understand this library for MS2. Is that theoretical or not?
Yes, or you acquire the data, you have this information. Yes. Yes.
So that is possible that the intensity of the heavy is not the same as the intensity of the light, or you find different?
The intensity, this is, yeah, it can be, of course, it's the, yes. No, the response is the same. The final intensity can be different because the heavy you will always have, and the light maybe in some samples. Yes, the response is the same.
It's the same.
Yes. Yes. Okay, so now we are saying that we were comparing this MS2 with our intensities of our fragments. Of course, like Joffre was asking, it has to do on how we fragment this. There are different types of fragmentation. We have seen CID, HCD. Each instrument varies a little bit. So it can vary a little bit, this rank of intensities, but it cannot be completely different. Okay, sometimes it's not exact. This is one publication that compares the fragmentation in the triple quadruple with these two different other types of fragmentation, which is CID and HCD. And the point of this paper was that HCD resembles more the fragmentation in a triple quadruple than CID. In this figure, they also put an extreme case. In general, they look quite similar, CID also with triple quadruples. But it's true that HCD maybe is the method of choice when you go, for example, to an Orbitrap to make your spectral libraries, is the method of choice to be able to compare later with the triple quadruple. But in general, the way the peptide fragments is quite similar. Okay, so are we sure we have our favorite protein and this is the one we are measuring? put a heavy standard in your sample, check these parameters we have been talking about, and never forget about measuring a unique peptide. So the last topic of this talk is about quantification. Now we know that we measure what we want to measure because we have the tools to see if this is right, okay? We have our heavy standard and we know that where the heavy is, our peptide is there. Are we quantifying? Because many times the point of the SRM is to have the correct, precise, accurate quantification. Are we doing this right? We need to think a lot about this, okay? It all depends on you, on how you are doing your things and how precise you want to be. There are two things to think about, the sample preparation and the SRM acquisition. Okay? In the sample preparation, you have all these steps, lysing your cells, trypsinizing your sample, maybe you do prefractionation or not, chromatography, and each one of these steps, there is a potential sample loss. And remember that in general, we go here and we put our heavy standard. And we're going to say that the amount we see, based on this heavy standard, is we're going to relate this with the protein that we had at the beginning. But the heavy standard, we haven't lost anything. And our endogenous protein, many things can happen to it. So we need to think, you know, in each step. One very important issue is the digestion efficiency. Okay, the digestion efficiency, which means which percentage has been digested, or how does it relate the amount of peptide that we have to the protein before digestion, is peptide dependent. So this is another issue about testing, about choosing our peptides. Okay, not each peptide represents as well our protein. And how do we know this? Testing, optimizing. It all depends what is the goal of our initial question, what is we want perfection, or we just want to have a quantification in different conditions and that's it. But this is something to think about. Some peptides represent better the amount of initial protein than others.
But if you know efficiency in each of these steps, you can come back to the
Exactly. Exactly. Another issue is peptide stability. We buy these heavy peptides, we have them in the freezer, and some, we test them 1 year later and they have the exact response. They haven't been degraded, everything is perfect. Some tend to precipitate, get more attached to the materials that they go through. They are less stable. So this is another thing that we can do, you know, a test to see the stability of our peptides. It, again, all depends how far we want to go. One good idea is always to include quality controls in each one of your steps, and add the internal standard as early as possible to account for this sample loss. Okay?
Stability of the standard is more important if it's absolute quantification.
Yes, exactly. If you are doing relative quantification, you are just comparing samples. But if you want to know the concentration of the protein in your sample, then you need to go much slower.
And the QCs, you do them with the proteins, not with the
The other thing is that instead of using the heavy peptide, the same peptide you're going to monitor heavy, some people do other strategies to try to avoid this problem. That is, for example, use proteins isotopically labeled and then digest them. There is one strategy that is the Q-concat, that what they do is that they concatenate all the peptides of the different proteins they want to monitor and then digest them to try to mimic a little bit more the real situation. There are different strategies. The simple one is to buy your heavy peptide. Or some people, what they do is they put their heavy peptide with the flanking residues of the protein to digest it and then account for all these losses, you know?
But even if the efficiency of the digestion is low, it's like 20%, 10%. If you compare different samples, they should be the same.
Yes.
So it should be corrected for that.
Yes. Yes, it's more about absolute quantification. The other issue is the way you acquire your data in SRM. Okay, you need to think about the instrument performance, your cycle time, and like always, having quality controls. What happens with the instrument? You can measure your protein in your samples, and you can have, you know, a curve like this. Intensity, time goes down, okay? If you add your quality control, and you see that it's behaving the same. Okay, so make sure you are measuring what you want to measure. It's not that in each sample you have less analyte, it's that the instrument is losing sensitivity.
And in how long does it take to lose all that sensitivity?
This is something that has to be checked, and you have to put quality controls in the middle, because then you can normalize. But the instrument can slowly, in months, lose sensitivity, or from today to tomorrow, you get a charge effect inside the instrument and it drops. So it's always monitoring this. Because if you happen to measure first all your healthy patients and then your disease patients, of course you're going to get the wrong conclusions. But this is also affects the experimental design. You can never do first all the healthy and then all the disease. Okay, this is going to be one of the topics tomorrow in the experimental design and statistics. But these are the things to take into account when you are doing quantification.
Then you need to be blind from the samples?
Tomorrow we're going to talk about this. Sometimes it's better not to be blind, but to know what you are measuring, so you don't get a bias of your measure. Tomorrow...
By not being blind?
Yes, because if you have 10 patients and you put them randomly, it can happen that you put first 4 healthy, then 5 disease, and then 1 healthy. And the last healthy, oh, this is an outlier. I still see a difference between healthy and disease that is not true.
Can you go back, please? Also, is the quality control of the instrument you do every day or every?
Yes. So in between samples, we run quality controls that, at least if the instrument is losing sensitivity, we know. So we can either stop and start over, or we can use this to normalize our data. Okay, and then comes the cycle time, extremely important. Okay, just 5 minutes of attention for the cycle time. Okay, we already talked about what is the cycle time. So if this is one signal of our chromatography, okay, we know that actually it's not continuous, but that we have points in which we measure. And we use the rest of the time to measure the other points. What happens if we only measure 4 points across our chromatographic curve? It can lead to wrong conclusions, because then we join these points in each case. We look at the area under the curve, and while this is the theoretical right amount, we see differences between the 2 groups which are not true. So our goal is, for quantification, always have at least 10 points across the chromatographic peak. And this is going to be closer, reproduce the real intensity of our signal. We always have to keep this in mind. If you have 3 points, you can identify your protein, but you cannot quantify it. How do we get these 10 points? This will depend on our chromatography, because the peaks can be narrower or wider, depending on our gradient, on the size of the particle, the column length. Okay? Longer columns, slower gradients separate better peaks, so we got better resolution, but they also widen the peak. So what is important is that we run our samples or some tests, and we know our chromatography. How does our chromatography looks like? And then we can decide, we want those 10 points inside our peak. Okay, these are real examples also. This is a column of 12 centimeters and 3 micrometer particle, and this one is a longer column with smaller particles. So the width of the peak, okay, goes from 21 seconds to 12 seconds. But we want to measure 10 points. It doesn't matter how many seconds we have, we want to measure 10 points. So we need to adjust for that. And this is going to determine our cycle time. Because the cycle time, this time that we have to do all the measures we want to measure or we would like to measure, it goes from this point to the next point. So here we have more time. We can measure more transitions than here. On the other hand, this chromatography is going to be better because we have a longer column and smaller particles. So it's a balance. But the most important thing is that we know our chromatography. And if we change the conditions, we need to study again our chromatography and adapt. Okay, so here is this exercise. No? Now we know that the number of transitions we can monitor in one single run is limited, and is limited by our cycle time. Okay, so say that we have a chromatographic peak that takes 20 seconds. How many transitions can we measure? If we measure 10 points, we will have a 2-second cycle time. And if we spend 20 milliseconds, that is what is called the dwell time, in each one of our transitions to measure it, we end up being able to measure 100 transitions. And how many proteins can we measure? Well, this depends on how many peptides we measure. In 100 transitions, if we decide to measure 4 transitions per peptide, 100 transitions translates into 25 peptides. And with 25 peptides, we can measure either 25 proteins with 1 peptide, or we can decide and maybe measure 10, 15 proteins. But we always need to know our chromatography, our cycle time, and from here we can make these decisions. These are 3 papers that I put here for all these thoughts that we have about quantifying correctly. They do these multi-center experiments with all quality controls that one can imagine, and these are nice papers to read if you are interested in doing one of these essays. Okay, so these are just some thoughts to remember about this talk. SRM is measuring triple quadruples, is the gold standard for quantification, use heavy peptides, know your chromatography, use quality controls.
If I wanted to measure 90 proteins, it's possible, but needs more time. That's the only.
Yes. In the practical, a little bit more practical talk after the coffee, we will see this. But you have 2 options. Do more runs in one same sample. If instead of running it 1 time your sample, you run it 3 times and you measure more things. Or you can do, that is what people do, scheduled SRM, in which you only monitor the transitions in the minutes that you know they are going to come out. So you are allowing more time and you can measure more things. Instead of measuring during 60 minutes, imagine your gradient is 60 minutes. Instead of for 60 minutes, you are measuring all your transitions. If you know that one peptide comes out in minute 10 and the other one minute 30 and one minute 60, you use this thing that you know because you already tested your sample, and you only use within that window of minutes, you measure the things that come out there. And then you can multiplex a lot more.
You have to be lucky that your peptides come in different times.
They always come in different times, but they always, exactly, but there is always an area, a time frame in which most come out. And that is going to be your limitation.
Okay, so you have to make a lot of tests before you come with an assay that works for your machine, ideally, and maybe not for my neighbor.
Yeah, ideally you test your things in your own hands, in your own instruments, and etc.
Because did they compare, I don't know if they did that already, like the inter...
Yes, yes,
assay,
yes. I don't know if it's one of these, but yes, there have been quite a few papers doing multicentric experiments. Let me see. No, this one, no. This one.
Okay.
This one is super recent, but it's not the only one. There were previous papers, but these ones are specific, especially careful about all these little things. What is the response of your peptide, stability, digestion efficiency. So these are like very thorough studies.
Is needed to put a routine method for diagnosis or no, to do this kind of thing?
What one of these, this one explains is like it puts different tiers or different, types of experiments, Do you want to set up a test for a clinical diagnosis? Then you need to go to the most precise, accurate. Are you asking about what are the differences in, or are you, I don't know, for example, looking at the interactors that you have depending on the cell conditions? This is a different case. You don't need to go exactly measuring all the peptides stability for 1 year. So they make you think about, you can try to reach perfection. It depends on what you want. And this one talks about this, and it's very nice.
And to get an absolute value, you basically use your heavy standard as a concentration.
You can get an absolute value, and we will explain this also in the next session, as long as you know the exact concentration of your heavy. And as long as you really take care of all these issues.
When you talk about, in the previous example, you say 25 peptides. Is 25 peptides or 12 plus the heavy, no?
Correct.
So
correct.
Double, no?
Correct. Correct. If you count that you have to monitor the peptide and the heavy, then the numbers get divided by 2.